Hello again. We're going to finish this chapter up strong, talking about the plasma membrane of the cell, and then we're going to talk about some internal structures that are really important. So the cell membrane is a selectively permeable membrane, just like in eukaryotic cells. We have hydrophilic heads. So these are the glycerol heads that are hydrophilic. And then we have hydrophobic tails. So if you don't remember what hydrophilic means, it means water-loving. Hydrophobic means water-hating. So the tails are situated towards each other. The heads are going to interact with the extracellular fluids and the intracellular fluids. Within the plasma membrane, we have a lot of proteins, integral and peripheral proteins. Integral proteins span the entire plasma membrane, whereas peripheral proteins are going to be found on one side of the membrane or the other only. Proteins are going to have a lot of different functions: movement of molecules, enzymatic reactions, metabolic reactions, where it's going to help in making energy. The cell wall has a lot of functions as well. So proteins have their own functions, but the cell wall has its own functions too. Whereas we have organelles in our body that have different functions, like we have the mitochondria that produces energy. We have the Golgi apparatus that acts as our post office that sorts and sends out different protein molecules. The plasma membrane is where energy production occurs. It's where molecules move into or out of our cells. It's where different metabolic reactions occur. And so here you're seeing facilitative transport, where a molecule binds, and then once it binds, the molecule or the protein opens up to the internal environment, allowing molecules that normally couldn't go through the plasma membrane to get through. So that's one function of the plasma membrane. Inside the cell, we have a structure known as the nucleoid region. The nucleoid region is where we hold the majority of our DNA. So I should say the majority of the bacterial DNA. Bacterial DNA typically is one single chromosome. It is circular in structure, and it's found in that nucleoid region. But sometimes bacteria also have, or I should say most of the time, bacteria also have extra chromosomal DNA. So not just the DNA found in the chromosome, they have structures called plasmids. Plasmids are smaller structures. They're also rings of DNA, but they don't they don't carry information that is mandatory for the cell to survive. What they do is they carry information that can potentially give beneficial benefits to the cell to make the cell more likely to be able to survive in the environment. Examples, some plasmids will give bacteria the ability to use certain forms of carbon that they normally couldn't use. Other plasmids might contain genes that allow them to survive in the presence of specific antibiotics. Bacteria have their own type of ribosomes. Ribosomes are the structures used for protein synthesis, so they're kind of like the workhorse. They sit there and they produce proteins all day long. That's their job. There are two units for a ribosome. A ribosome has a large subunit and a small subunit. Bacterial ribosomes are 70S ribosomes. That's what they're known as. but they consist of a 50S subunit and a 30S subunit. So we don't add the subunits up to get the full ribosome. Eukaryotic cells have their own ribosomes. Eukaryotes have 80S ribosomes, consisting of 60S subunits and 40S subunits. So always remember that 70S ribosomes are bacterial, 80S ribosomes are eukaryotic, but 70S ribosomes consist of 50S and 30S. Okay. Microcompartments. Most bacteria have specific regions where they can carry nutrients or where they might carry gases to allow bacteria to move in environments. Specific bacteria contain magnetosomes, which are tiny little magnet, basically, magnetic material that allows them to recognize the different magnetic fields in the earth. And so when you think of gas-filled vesicles, think about when bacteria want to gain nutrients, they might have to release air or gas to become more buoyant and float upwards, or they might take on more gas so that they can fall down to either gain nutrients or to get away from predators. Bacteria have their own cytoskeletal proteins. They're very similar to eukaryotic cytoskeletal proteins. They function the same way. They help to move materials around in the cell. They help to regulate cell division, and they help in determination of the bacterial shape. Some bacteria are able to produce endospores. Endospores are special cells that allow bacteria to survive in environments that are not conducive for survival. So the two groups of bacteria that we talk about that produce endospores, and the two main groups are Bacillus and Clostridium. We only use Bacillus in our lab. Clostridium we have tried, but they are obligate anaerobic bacteria, which means they cannot replicate in the presence of oxygen, and we have not been successful in growing them. Bacillus, on the other hand, is able to grow in oxygen, so it grows well. Bacillus can lie dormant for hundreds of years. And then if the environmental conditions become beneficial, the endospores can germinate and become normal vegetative cells. So endospores are formed when the environment becomes less beneficial. So when there are not enough nutrients, oxygen, the agar is very dehydrated, whatever the environment is like that makes the environment not beneficial to life, then the bacteria will start to develop an endospore. So it will go through the normal replication process where DNA are replicated, but then we produce a spore, and that spore, so basically we produce a tiny little split between the DNA, so it replicates, and we form this little barrier between the two strands of the two chromosomes. One of the cells, the larger of the two cells, starts to engulf the smaller of the two cells, forming a spore. Then we have different molecules enter to dehydrate the entire cell, the spore. We have peptidoglycan laid down in between the cell wall, and then the parent cell will basically degenerate. And so you'll have this nice thick spore surrounded, endospore surrounded structure that is not a newly replicated cell. It's the same cell that was there, but it's just went into a dormant kind of hibernation.
Spore-forming bacteria grow as vegetative cells and divide by binary fission when nutrients are available and environmental conditions are not adverse. When nutrients are depleted or conditions become adverse, spore formation is initiated. The DNA condenses and aligns itself in the center of the cell. The vegetative cell is now referred to as the mother cell. Next, the DNA divides into two complete copies, and the mother cell membrane invaginates to form the developing forespore. The mother cell membrane continues to grow and engulfs the developing spore. The developing spore is now surrounded by two membrane layers. Next, peptidoglycan is laid down between the two membranes of the developing spore to form the cortex. Dipicolenic acid is formed inside the developing spore, and calcium enters from the outside. As calcium enters the spore, water is removed. A protein coat forms exterior to the cortex, and the spore becomes mature. Some spores form an additional layer called the exosporium. A mature spore is resistant to environmental conditions. Finally, lytic enzymes destroy the mother cell and the mature spore is released.
All right, so here's a question that you can think about. It's not something I'm going to answer here. It's something that I want you to think about, and if you're not sure how to answer it, bring the question to me. Wait, there's another one. Both of these I actually did answer in our lecture today, either the first one or the second one, okay? I'm going to stop here, and I will see you on Tuesday. Bye.